Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Tarek O'Dali from Business Review, and I will be your host. It is our pleasure to have BioPharmaSpec with us today, who will be presenting this webinar titled Structural Comparability Studies for Biosimilars. Today's guest speaker is Dr. Richard Easton, Technical Director, Structural Analysis. You'll be hearing from him in a minute, but first I'd like to welcome you to our webinar platform. This is On24. You'll notice it's fully browser-based. So if you do find yourself disconnecting for any reason, please just click on that link that you received via email. You should be able to rejoin the session immediately. In order to ask questions, you can send them in via the questions widget. So type your question into the box there at the top left-hand corner of the screen, then click Submit. We're going to try to allocate some time at the end of the session to address any questions or thoughts that you may have. If you click on the green resource list widget at the bottom of your screen, you'll be able to view or download some resources that are relevant to this webinar. Also, if you find yourself in need of technical assistance, you can always use the yellow help widget at the bottom there as well. You are free also to move, resize, and maximize any of the windows in front of you to get a better view of the slides, and you also have the option of going full screen as well. But now, housekeeping out of the way, please allow me to welcome today's speaker. So Richard, over to you.
Thank you very much, Tarek, and hello to everybody. Thank you all for taking time out of your schedules to join our webinar today. As mentioned, I'll be talking about the structural comparability studies for biosimilars. I'll talk first of all a little bit about who we are and what we do at BioPharmaSpec, and then I'll get into the main body of our presentation. So first of all, BioPharmaSpec is a contract research organization. We are focused very much in the preclinical area. We're 4 years old as a company. Our founder was also the founder of the MScan group of companies in the late 70s. That's Professor Howard Morris. And many of us, myself included, and our MD, worked for MScan for many years before we moved to BioPharmaSpec. This means that we do have extensive understanding of the biopharmaceutical characterization. We use state-of-the-art mass spectrometric instrumentation, along with chromatographic instrumentation and other techniques, to perform a full range of structural characterization studies. The experience that we have gained over many years of pioneering work, both in academic and industrial areas, is directly applicable to the work that we perform. We've been involved in the development and the setup, really, of techniques such as peptide mapping, disulfide bridge analysis, glycan analysis, and Professor Morris himself was involved in the conceptualization of the QTOF mass spectrometer, which is now a cornerstone of analytical studies in this area. We have two labs, one based in Jersey in the Channel Islands, that's our UK lab, and one in the USA in Pennsylvania. We have a lot of experience within the biosimilar analysis area. There's a list on the screen here of just several of the molecules we've worked on. There's a much more extensive list. A lot of these, as you can see, are monoclonal antibodies, but also there's a range of other molecules that we've worked on, such as fusion proteins, an example of which would be etanercept, heavily glycosylated molecules, such as erythropoietin and its related products, and other derivatives, such as pegylated GCSF-type molecules. So we have a lot of experience in both comparative analyses and general analyses of biosimilars and other biologics as well. So why do we actually have to do these characterizations for biosimilars? Well, of course, the clue is in the name, really. It has to be biologically similar to the molecule that we're comparing it to and that we're claiming it's a biosimilar of. This means that a large body of structural characterization and biological comparative studies have to be performed. We'll be focusing on very much the structural characterization today. The documentation for this is very thorough, as you can imagine. The regulatory agencies have brought out very clear guidelines now as to why and how we go about performing these structural studies. I'm showing some examples here from the EMA and also from the FDA, but this applies much more globally as well. So other regulatory authorities are saying basically the same thing. If we just take a look at the EMA guidelines, and this is their quality issues guidelines for biosimilars, they talk about the use of extensive state-of-the-art characterization studies that need to be applied to the biosimilar and the reference product in parallel. And they specifically state that ICH Q6B is the reference text to look back to for what these techniques and methods are. And we will be talking about the ICH Q6B guidelines in quite some detail in a little while. If we look at the equivalent FDA scientific document for biosimilars, they talk about using techniques with adequate sensitivity and specificity, and they go on to list particular areas that need to be looked at: primary structure, higher order structure, post-translational modifications. They mention glycosylation and phosphorylation as examples, but all other modifications need to be examined. Other possible variations, such as deamidation and oxidation, which can arise through issues with maybe purification or storage of the sample. So these are more chemical modifications that are occurring outside of maybe the natural product's range, as well as other specific modifications or intentional modifications, such as pegylation of molecules, if that's relevant. And this is effectively ICH Q6B in all but name. If we look at their sister document, the quality considerations, the FDA particularly talks about the analytical data being the foundation of a biosimilar development program, and that this can then lead to decision influencing for the subsequent clinical data that is required. They go on to say that analytical data should be available early in product development, and they actively encourage the submission of this data at an early stage, and they can make decisions once they've assessed this analytical similarity data. So there's very strong emphasis on producing data early, producing it thoroughly, and producing it such that subsequent work can be directed in an efficient manner. So know your molecule well and know it soon. Okay, and with that, we'll pass on to our first poll question, and I'll hand back to Tarek for that.
Thank you very much. So yes, this is an interactive polling question. Audience members, please do take part. You just need to answer the question there by selecting one of the options and clicking submit. The question is, where do biosimilars sit within your current product portfolio? And the options you have: we are focusing heavily on biosimilar development in our current product pipeline. Option 2, biosimilar development is only a small part of our current product pipeline. Number 3, we are not developing a biosimilar yet, but plan to in the near future. And number 4, no current plans for a biosimilar development. So please go ahead and select whichever one most applies to you and click submit. And while you do that, I'm just going to hand back over to Richard briefly just to talk a bit more about this question.
Okay, thank you, Tarek. Yes, so this is going to be quite an interesting one to see where people sit. My expectation is that we'd see one of the first two options being the bulk of the vote, but it will be interesting to see. Obviously, different people have different ideas about how to approach this, whether they are going to focus very heavily on it or put that as part of a package, but not necessarily rely very heavily on that. There's a lot of biosimilars that are out there in the market, so I think it will be interesting to see just how people are positioning themselves with regards to the biosimilar product versus maybe innovative products within their own portfolios.
Absolutely, very interesting. So let's have a look at the results now. Thank you very much to everybody who took part. And okay, so it looks like the majority of the audience there have gone for option 1. We are focusing heavily on biosimilar development in our current product pipeline. What do you think about that result?
Yes, that doesn't surprise me. They are a very large area of the current global product. It does make sense that people are looking to focus on those. There's a lot of different products that will be coming off of patent, and people are looking to get involved in those. So yes, that does make sense to me. Yes. Okay, thank you everybody for taking part on that. We'll move on now. So what I'll talk about now really is just to give a little bit of an idea of how and when the analytics can be brought into play and where different levels of analysis can take part or take place within the kind of sort of flow diagram of product development. We will be focusing mainly on comparability, but it's also worth bearing in mind, or it's very important, I should say, to bear in mind, that setting the control of structural characterization, that can really take place as soon as the idea of which biosimilar is going to be manufactured has been put into the system. So as soon as you know what you're going to be working on, it makes sense to start whatever characterization studies are pertinent as soon as possible. To that end, the most important thing to perform is the primary sequencing of the reference medicinal product or innovator. This really can take place as soon as you know what product it is that you're going to be making the biosimilar of, because obviously you have to show that the sequence of your molecule is the same as the biosimilar, same as the innovator, I'm sorry. Now that is again something that's clearly documented, and we'll show that in a second. But what this does mean is that you need to know exactly what is on the market at the current time in terms of what its sequence is. So as soon as you know what you're going to be working on, get a batch of that innovator material and perform some sequencing studies at the amino acid level, not just DNA, but at the amino acid level, to determine the nature of the structure of the molecule on the market. It's not enough to know what the DNA sequence is, and also it's not enough to know what the published sequence is, because there could be errors within the published sequence. You need to know and need to generate data from batches that are currently on the market. And you only need to do it on one batch, but you know exactly then what the structure is that you are targeting. So you can do this before any biosimilar is actually made. And obviously this shows that you will then be working on the correct sequence when you ultimately go and produce your molecule. Subsequently, as you start to produce material or start to do various things such as clone selection, you can then focus on particular analytical approaches that will be key to producing a molecule that has the same target product profile as the RMP. So in other words, right from the outset, you should also be looking at various batches of product that are on the market to see what they are structurally like, so that you have a target to focus on when you actually produce your molecule. So again, you can be constantly assessing batches for the various structures such as glycosylation, if it's relevant to your molecule, the peptide map, the intact mass, looking at these key features of that particular molecule to make sure that when you produce your molecule, it sits nicely within that target. To this end, clone selection is obviously very important, and there will be key tests that you need to perform on these clones to find the most appropriate one to take forward to ultimately produce much more detailed work as you subsequently produce ultimately the biosimilar itself. Purification is actually very important as far as this goes. Now, once you've actually produced your biosimilar, it will be in its own formulation. However, a lot of these formulations can have an impact on the actual analyses themselves. The regulatory authorities understand this, and they know that it is quite acceptable to purify the molecule out from its formulation to allow for analysis. However, if you are doing that, then you need to show that that purification is not having an adverse effect on the removal of any components that you may need to examine. So, for example, if there are possibly truncated species of your molecule that are present, you need to show that you're not purifying them away, and any subsequent tests you do therefore show an absence of them, when in fact they're present, but they've fallen through the gaps of the purification process. So the purification is very important. It needs to be shown to be robust for the molecule and to not interfere with the key structural elements of the molecule or any modified forms of it in any way. Once you have this method in place, you can then use that for your subsequent characterization. Also, once you have a batch available of your biosimilar, it's important to carry out the amino acid sequencing in exactly the same way as you did for the RMP. You need to show that your molecule is identical at the amino acid level, and obviously you certainly hope that it is. If not, then it's back to the drawing board to find out what went wrong. The extinction coefficient is another very important element of the molecule to get a handle on quite early on. The reason for this is that you need to know how much material you're actually handling if you're going to consider the efficacy and the safety as well as the quality of the product. So determining the extinction coefficient experimentally is very, very important. It's not enough to determine it theoretically, since the overall structure of the molecule can mean that the different amino acids and their local environment can affect what the theoretical extinction coefficient of that amino acid would be. So you have to actually determine this experimentally now, and there is an expectation that that is done. Theoretical is no longer sufficient for this. So finally, you can actually perform the comparability itself. Obviously, you need to show that the biosimilar is structurally comparable to the reference material. We need to perform a full structural assessment as outlined in topic Q6B of the ICH guidelines. This will cover both the primary structure as well as the broader, higher order structure and physicochemical properties, and we'll talk all about those shortly. The number of batches that need to be analysed is product dependent. There's no set number for this, so you may well need to get guidance on that. The reference material that you use for the comparability studies, it's important also to use this from different stages of its expiry, or rather its life cycle, and also from different manufacturing sites. That will give you the fullest breadth of structural information on that RMP and the largest target to hit effectively. So if we go back to our guidelines and have a look at what they say, if we look at the EMA guidelines, their quality issues again, they say in section 5.1, multiple different batches of the reference product should be used to provide robust comparability data in order to generate a representative quality profile. Again, no mention of batches. FDA likewise, in their scientific considerations for biosimilarity, the proposed product and the reference product in multiple representative lots to understand the lot-to-lot variability of both products. Again, there's no mention of how many batches to examine in there. In broad terms, you would probably be looking at somewhere between 12 and 20 batches on a comparative scale. That's a side-by-side analysis to show how similar the molecules are. But of course, that will be dependent on the product and may well be dependent on the data that you generate at each time point, or rather at each comparability set. So you will need to obtain guidance from the agencies on exactly what they're expecting from your particular product. Okay, so we know that this ICH Q6B document is referenced. We know that there is an expectation for full characterization using it, but what exactly does it entail? Well, I've listed some of the points here. And just to talk about what this is, the ICH is the International Conference on Harmonization, now the International Council on Harmonization. This is a body of regulatory authorities and other groups that have got together to produce a series of harmonized guidelines for what they expect to be performed in terms of producing biological and indeed other products as well, other pharmaceutical products. Initially, it was the EMA, the US FDA, and the Japanese regulatory authorities that got together for this work. Subsequently, many other groups have now joined that. Sorry, the slide just disappeared there. The document Q6B is a specific document for talking about the structural characterization of these biological molecules. Within that, in Appendix 6.1, there is a list of various different areas that need to be considered as far as the analytics go. This is broken down into two broad sections. First of all, we have the structural characterization and confirmation. This is very much looking at the primary level of the molecule. So we carry out work on the amino acid sequencing, as well as post-translational modifications, looking at disulfides and carbohydrate structure, for example. The physicochemical properties deal more with the broader gross features of the molecule themselves, as well as higher order structures. Now, we don't have time today to go into all of those, I'm afraid, but we will be covering the areas highlighted in blue. But before we do that, I'd just like to move on to the next poll question, so I'll pass back to Tarek for that.
Thank you very much, Richard. Now, audience members, please make sure you read through the options here and select any that apply and hit submit. It's another interactive polling question. So the question is, if you are developing biosimilars, which of the following are in the preclinical or phase 1 stage? So have a look at those options there and make sure you select any that apply and then hit that submit button. While you make your selections, I will just remind you as well that we will be having a Q&A session at the end of today's session, so please make sure you, if you have any questions, please make sure you submit those. The box for that is at the top left of your screen. And I'm just going to hand back to Richard very briefly just to talk a bit more about the options on the screen and about the question as well. So Richard, what do you think the result is going to be here?
Again, what we've done is chosen various different categories that encapsulate, broadly speaking, quite large areas of biosimilar work. I would suspect that the monoclonals and ADCs would be the bulk of this, but obviously we're only talking about the preclinical area here. I would expect that to be the largest, probably with the fusion products and molecules such as the erythropoietin type molecules to be up there as well, maybe coming in at second. But it will be interesting to see what people come back with on that.
Absolutely. Audience members, if you still have not voted, please make sure you do. You do need to click the submit button to submit your vote. I'll just give you a few more seconds to do that now, and then we'll go on and have a look at the result. The question, once again, if you are developing biosimilars, which of the following are in the preclinical or phase 1 stage? Okay, I think that's given you plenty of time to take part. Thank you very much to everybody who has. And now let's have a look at the results here. So, okay, so it looks like the majority of the audience there going for this option, monoclonal antibodies and ADCs. What do you think of that?
I'm not surprised that is the most abundant one. That does make sense. They're a very large area for biosimilar work. So 75%, that's expected. I would have thought that the fusion products and erythropoietin type molecules would have featured slightly higher than they have. So that's an interesting result, actually. The monoclonal antibodies is way ahead of everything else. Yeah, interesting. Okay, thank you everybody for taking part in that. We'll move on now. So we've talked about the need for amino acid sequencing. The ICH guidelines list amino acid sequence information as the first topic on there. Now, the ICH itself, when this was set up, the techniques that are available now were not available then in terms of their ability to sequence molecules. So sequencing really came in from a collection of other techniques such as peptide mapping and amino acid analysis. Nowadays, the equipment and instrumentation and techniques are available to perform amino acid sequencing on molecules as large as monoclonal antibodies in its entirety, and there is an expectation that that will be performed. Once again, looking at the guidelines, if we look at the EMA guidelines, their quality issues document specifically says the target amino acid sequence of the biosimilar should be confirmed and is expected to be the same as for the reference medicinal product. So there's no room for misunderstanding there. Full sequencing is needed at the amino acid level. This is also true of the FDA. Their scientific document for biosimilars specifically states it's expected that the expression construct for a proposed product will encode the same primary amino acid sequence as its reference product. Once again, the expectation is that full amino acid sequencing is done, and this specifically states here state-of-the-art technology as well. So the authorities are well aware of the techniques and instrumentation that are out there, and they expect these to be applied at the amino acid level. So how do we go about doing that? Well, there are two main procedures for this. Each procedure has an advantage and a disadvantage, but working together they can provide full sequencing. And these two procedures are so-called gas phase sequencing using Edman degradation, which is a chemical procedure. And the second procedure is the mass spectrometric sequencing approach, and we'll talk about both of those. So if we just look at the MS sequencing, first of all, this is performed by taking the sample, taking the molecule, performing a series of different proteolytic digests, enzymic digests, to break the molecule up into peptides, and then using an online liquid chromatography with electrospray mass spectrometry. That's the ionization technique, electrospray, with either so-called MS to the E, which is a Waters, the instrument manufacturer, specific technique for generating fragment ions from peptides, or the so-called data-dependent acquisition method for selecting a particular peptide and then producing fragment ions from that. Either way, the idea is to produce a series of fragment ions from each of those peptides, and because the fragmentation occurs at the amide backbone, in other words, the link between the individual amino acids, because amino acids have different masses, broadly speaking, we can look at the mass differences between the fragment ions to work out what the different amino acids are. And we can actually sequence each peptide and the amino acids of each peptide in that way. Now, if we use multiple digests, we can identify regions in other peptide digests that we cannot necessarily see in one digest, because it may be that they're too small or too big. And it also means that we can generate regions of sequence that we can stitch together and therefore generate overlapping peptides that will ultimately lead to sequencing of the whole molecule. However, there are amino acids such as leucine and isoleucine that are so-called isobaric amino acids. They are isomers of one another. They have exactly the same mass. So you cannot distinguish in a mass spectrometric sense what these different amino acids are. You'll see a particular mass difference, but you don't know whether it's one or the other. In instances like that, what we can do is collect those peptides that contain these unassigned signals, or we can perform the N-terminal sequencing, the Edmond sequencing, to chemically release one amino acid at a time from the N-terminus of the molecule, each amino acid, and we can identify them by their run positions on a chromatogram. And the leucine and the isoleucine derivatives will run at unique positions, and thus we can say precisely which one is which. Now, the Edmond chemistry does suffer from the fact that there is a background that builds up over a number of cycles because the yield is not 100%. So that does mean that after a while you will get background that makes it hard to distinguish the amino acid you're releasing. Furthermore, if you have a blocked N-terminus, such as a pyroglutamate or an N-acetylated terminus, then the Edmond chemistry doesn't work. It does need a free terminus. But a combination of these two techniques does allow you to sequence fully a molecule even as large as a monoclonal antibody. ICH also talks about peptide mapping. Now, this is a very powerful technique. We use mass spectrometry to generate a series of peptides in this way. The procedure involves taking a molecule. Here we have a monoclonal. Cleaning it up if necessary to remove those buffer excipients. We talked about purification earlier. We reduce the molecule to produce a series of chains. We carry out specific protease digest to produce peptides of known size. We separate them out by online LCMS, and we can identify those different peptides by their masses and confirm that those masses are exactly as we would expect for the molecule based on the sequence that we've generated. Now, here we're not looking to look at all the fragment ions, but we're looking to see some fragment ions that will confirm what that peptide is. Now, because this is a mass-based analysis, it does mean that anything that modifies the mass is also going to be detected. So, for example, post-translational modifications such as oxidation and deamidation can readily be identified. Glycosylation can be readily identified. Disulfide bridges can be assessed by absence of a reduction step in the initial preparation. So there's a whole range of different structural features in the molecule that can be assessed through the peptide map. It's a very, very powerful tool. An example of the kind of data that we generate, if you compare a reference product with biosimilars, for example, is shown here. So here we have the reference product at the top and 3 different biosimilar batches underneath it. And visually, at first glance, they look pretty comparable, but if you look closely, you can see this peak at about 11.5 minutes is different between the reference product and batches 2 and 3. Batch 1 is looking identical. So on this basis, depending on what batches 2 and 3 have or how they've been produced, you would either change that manufacturing procedure, or you would eliminate those kind of batches and just proceed with your batch 1 for your biosimilar work. An example of the kind of data that we generate is shown a bit more clearly here. This is from a QTOF mass spectrometer. This is the instrument I mentioned earlier that our chairman, Professor Howard Morris, conceptualized. The trace you see at the bottom is the total ion chromatogram. That's effectively the overall data that is generated. The trace above is the so-called extracted ion chromatogram, where we specifically search for one mass, and that is the mass 660.4 Daltons. This is consistent with the C-terminal peptide of the heavy chain. So we have a peak at about 6-odd minutes there, 6-7 minutes. If we look at that more closely, as you can see here, the low energy spectrum shows our peak at 660. The high energy spectrum shows fragment ions that have derived from that, and that data shows fragment ions consistent with the C-terminal peptide as we would expect, and that confirms the identification of this molecule. And we can perform this kind of analysis throughout the spectrum and confirm the different peptides that are present, and should we see a mass modification, we can identify where that modification has taken place and potentially what it is, depending on that mass shift. Now, it is possible to look at glycans from a peptide map. However, you will not necessarily see all of the glycans present in there, and they can suffer from desilylation effects in the source of the instrument. So it is expected that the glycans will be removed and characterized separately. And again, the ICH Q6B document specifically talks about oligosaccharide profiling. Now, there's several different techniques that need to be used to carry out a full characterization package on the glycans, and it is expected that these are employed. But the only technique I want to talk about now, because of time, is the fluorescence analysis of the released glycans. So this is a specific procedure to get a chromatographic population profile of the N glycans. In this instance, we take our molecule, we reduce it again, we carry out a proteolytic digest to open the molecular structure up and produce a series of peptides. This is amenable to the enzyme PNGase-F, which is a D-N-glycosylating enzyme. It specifically releases the N-glycans from the glycopeptides. These can then be purified away using, for example, small C18 set pack cartridges, and those N-glycans can then be specifically fluorescence labeled and separated by hydrophilic chromatography, along with fluorescence analysis and mass spectrometric analysis of the products. This allows us to identify what the components are in the chromatogram based on their mass and their retention time. It is possible to circumvent to some extent the proteolytic digest. The use of modern buffers allows PNGase-F to work well on larger structures by opening it up, so that can bypass the need for the proteolysis step and speed things up. This is an example of the kind of data that we generate. Here you can see a glycan profile with the various glycans labeled on here from a monoclonal antibody. And you can see that the column separation is sufficiently good and sufficiently highly resolving that actually isomeric forms can be separated. So if you look at the structure G1FA and G1FB, they have specific elution times, but the only difference between those two structures is the location of that yellow circle, which is a galactose on either the upper or lower arm of that core structure. So if we take the data from our reference molecule that we saw earlier in the peptide map, and we compare that to the glycan data that we generated from the batch 1 biosimilar, again from that peptide map, you can see that this component, the so-called G0 component, does differ between the two structures, between the two molecules. Now, whether this has any effect in terms of the biological activity, that would require bioassay work to be performed. Nonetheless, that difference does exist. It has to be noted and it has to be understood. And that potentially is a difference that would not be seen in the peptide mapping. So release and analysis of N-glycans is really a key and fundamental part of structural characterization. And it does produce profiles that you can then compare one to another very readily. Okay, with that, we'll move on to our third poll question, and I'll pass back to Tarek.
Thank you very much, Richard. Audience members, once again, please select an answer from the ones below and hit submit. So the question is, which area of structural analysis do you find most challenging to perform? That's which area of structural analysis do you find most challenging to perform? Is it amino acid sequence conformation? Is it glycosylation? Post-translational modifications, for example, disulfide bridges? Higher order structural analysis, aggregation, or no challenges in any areas? So go ahead and have a look at those options. Find whichever one is the most challenging, that you find the most challenging to perform, and select it, and then hit submit. So we're going to have give you a moment for all the answers to come in, and then we'll have a look at the results. Before we do look at the results, Richard, do you want to comment briefly on this question before we move on?
Yes, well, it would certainly be interesting to see if people don't have any challenges in any areas. I suspect that won't get too many votes. All of these different areas that we've covered, they require different instrumentation, a different understanding of the molecule, and so the availability of different instrumentation could well be what the limiting factor is here. I would suspect that things like glycosylation, possibly higher order structural analysis, could be the most challenging. But yes, we'll see what people come back with on that.
Absolutely. I'll just give you guys a few more seconds to vote for this, and then we'll have a look at the results. The question once again: which area of structural analysis do you find most challenging to perform? Few more seconds now, and I think that's given you enough time. Thank you very much to everybody who voted, and let's have a look at the results. Okay, so it looks like it's quite a mix there, but the majority of the audience have gone for post-translational modifications, for example, disulfide bridges. What do you think about that, Richard?
Yes, well, this could be an interesting reflection of the number of people who are maybe working on things like monoclonal antibodies. The glycosylation is more limited there, so maybe that's not posing so many problems. Post-translational modifications themselves are very, very broad, so it's not surprising that maybe people do struggle with things like that as well. Higher order structure, again, that does require specific instrumentation. So that's maybe not wholly unexpected when you consider the kind of breakdown of biosimilars that we had in the earlier polls. Okay, thanks everybody again for voting on that. If we just continue now and start to look at other areas of the ICH Q6B, if we move into the more kind of physicochemical characterization, so this is the second set that we were talking about on the earlier slide. The first one that I want to cover is the intact molecular weight analysis. The idea behind this, of course, is that we see what the overall intact mass of the molecule is. This is aided by the fact that instruments such as the Q6B mass spectrometer are very sensitive, they're very highly resolving, and they're very mass accurate, which means even large molecules such as monoclonal antibodies can have their mass measured to a very high degree of accuracy indeed. And this means, of course, that any modifications that have altered the mass from what is expected will show up quite readily, and then this can lead to maybe particular assessments of particular parts of the structure, or indeed to revisiting things like the peptide map to see if the assignments or the theories behind why the mass could be different are reflected in some of the peptides that are detected. So we take our molecule, in this case a MAB is shown, we clean it up if needs be to remove excipients should they be interfering. We can, depending on what the buffer formulation is, of course, do this directly online, and that may well be feasible. The data shown here is from a monoclonal antibody purified online LCMS-wise using a Q6B mass spectrometer. We have a series of peaks present, as you can see there, at about 148 kilodaltons. Each of those peaks is separated by the mass of a galactose unit. So what we're looking at here is variation in the glycosylation of our monoclonal antibody, which is exactly what we would expect would be the case. But should there be any other clear molecular variation, we can go back and assess other data that we've generated to see if we have supportive evidence for that modification. So in other words, we're starting to generate orthogonality of data, and that's really what the authorities want: supportive, robust, strong data that is self-supporting. We can take our molecule, we can reduce it to produce separate chains if needs be. And we can analyze the free light chain, which should have, hopefully, the mass that we will predict, and the same for the heavy chain. And this helps confirm that the overall mass of the molecule is composed of those units that we would expect. And again, because the heavy chain carries the glycosylation, we see a series of peaks, each separated by the mass of a galactose unit. Okay, so that's intact mass. Another area we need to look at is the overall isoform pattern of the molecule. So we're looking at the charge profile of the molecule with this technique. At BioPharmaSpec, we use imaging capillary isoelectric focusing for this. This means that we can separate the molecule in the capillary and analyze everything within that capillary at the same time. So rather than have to pass everything through a detector, we can directly analyze what is in the capillary in real time. Just to give you an idea of some data from monoclonal antibodies, here we have one with a PI of around about 7.4. On the left is the full trace. You can see PI markers at the low and high pH points. And on the right, we've zoomed in on that. You can see the main component at about 7.4, and then two others at slightly more basic PI, about 7.5 and 7.55 odd. These are composed of the heavy chains carrying one or two C-terminal lysine residues, making the overall molecule slightly more basic. And again, we would expect to see this kind of variation in C-terminal lysine within the peptide map. If we take a second monoclonal antibody, this is a different one with a different PI. Again, overall trace shown on the left, and then the zoomed in data shown on the right. We have a main component around about 9.1. About 9.2, we have the more basic form. Again, variation in the C-terminal lysine, different to the previous one that we showed. But now we have two components at slightly more acidic PI values. This is consistent with deamidation within the molecule. These deamidated forms can then be assessed out of the peptide map to confirm that we see those, and therefore to act as supportive data for the data that we're generating from the IEF profiles. Another area we need to look at is the electrophoretic profiles. This shows capillary electrophoresis of monoclonal antibodies again. On the left, we have the non-reduced data, the intact data of a reference sample, and then other monoclonal antibodies around underneath, other biosimilars of that. And you can see a single peak in each case, one or two small bumps as well, but mainly a single peak. On the right, we have the reduced molecule showing the light and heavy chains. And again, this serves as supportive data for the intact mass that we've already generated. This is showing that, hopefully, there are no small fragments that are being released upon reduction, and that there's no other components that may exist within that sample. So supportive data, again, from a relatively simple technique, but nonetheless something that needs to be done and helps to bolster data from other sources. Spectroscopic profiles is another area that we need to look at. We at BioPharmaSpec use circular dichroism quite heavily as one of the techniques for analyzing secondary structure, and that's what we're talking about really with the spectroscopic profiles. It's an assessment of the higher order structure of the molecule. So here we're looking at secondary and tertiary structures. In the case of CD, what we're looking at is the differential absorption of left and right circularly polarized light. Now, when this is performed in the far UV region, we can observe secondary structural features. When this is performed in the near UV region, we can analyze tertiary structural features. So we, of course, do both to look for both secondary and tertiary structures. Now, in the case of the secondary structure, we can generate data from the innovator and from the biosimilar samples. The data can be passed through a particular algorithm that will measure the data generated and compare to a database of known molecules, and that will then lead to the production of a table showing the different percentages of the different forms of secondary molecular structure that are present, such as beta sheet, alpha helices, turns, for example. And obviously, we'd look to show that there's similarity between the biosimilar and the innovator. In terms of near UV for the tertiary structure, we're simply looking at the comparative profiles of the biosimilar versus innovator. And on the screen here, you can see a biosimilar and innovator molecules, and the profiles are more or less overlapping, as you would certainly hope. Now, we've already talked about the orthogonality and the importance of orthogonality. Spectroscopic profiles is no exception to this, and as an orthogonal technique, we use Fourier transform infrared analysis. In this case, what we're looking at is the various stretchings and bendings of bonds involved in the amide linkages through the molecule, and the precise wavelengths at which these bend or stretch or flex will depend on the local environment of the structures. So to that end, we can generate a profile using FTIR that will be reflective of the secondary structure, i.e. the local environment of these bonds throughout the molecule. And again, this data can then be converted to give percentages of the secondary structure, the alpha helices, the beta sheets, turns, etc. Now, the data from CD is not necessarily going to be exactly the same as the data from the FTIR because they're different techniques working in different ways. However, in terms of comparability, what we're looking at is to show that the data within each technique is comparable between the innovator and the biosimilar. So the final technique that I want to mention today is aggregation analysis. Now, aggregation doesn't specifically sit within the list of products, or rather of tests, within ICH Q6B, as I showed earlier. However, it is considered within there as a degradation product or an impurity, if you like. So there is a requirement to look at aggregation from the ICH Q6B perspective, but also there's specific mention of this within the biosimilar guidelines themselves. So once again, looking at the FDA guidelines, their quality considerations, they specifically talk about the degradation pathways, and they mention oxidation, deamidation, and aggregation. They talk about a comparative analytical characterization of these areas, and they specifically say that more than one analytical procedure needs to be performed, i.e. orthogonal methods to assess aggregation. So even here, we're looking at using different techniques to generate supportive data on this particular aspect. Likewise, the EMA guidelines, they once again talk about comparative analysis using a combination of analytical procedures to look at aggregation, amongst other things, from degradation pathways. So at BioPharmaSpec, we use orthogonal techniques for the aggregation analysis. The two techniques we use, I'll talk about now. The first one is SEC-MOLS. This is size exclusion chromatography with multi-angle laser light scattering. In this technique, we use ultraviolet, refractive index, and moles detectors to generate mass and relative quantitative information of the different size components present in the samples. This allows us to assess monomeric and multimeric states of the molecule. However, it is a column-based technique. So what that means is that the molecules will be drawn apart as they pass down the chromatography system, and that kind of shear effect can lead to the breakdown of some aggregates. So we can measure aggregation, but we may find there's a decreased concentration within the sample because of the nature of this column separation. Once again, because it's a comparative analysis tool, we're comparing the biosimilar to the innovator. So within this technique, we can generate perfectly acceptable comparative data. As an orthogonal technique, we use sedimentation velocity analytical ultracentrifugation. The FDA particularly like this because it's a column-free technique. Now, the theory behind this, of course, is that the sample is spun at very high speed, causing monomeric and multimeric species to separate out because they're different sedimentation coefficients, the different sizes within the system. Because this is a column-free technique and because we are effectively compressing the components together, we can be pushing the aggregation the other way. So we can actually be generating, to an extent, aggregation in this. So we can, once again, generate perfectly acceptable numbers that, from a comparative point of view, are absolutely fine and adequate and acceptable to the regulatory authorities. But the numbers within this technique may well be different to the numbers that you would generate from the SEC-MOLS analysis. Effectively, this is giving you an upper and a lower level for the degree of aggregates. But we are talking about comparability, so part of within the comparability exercise, these techniques are both perfectly fine and will generate data that is acceptable to the authorities. Okay, and with that, I will pass back to Tarek for our final poll question of today.
Thank you very much, Richard. Yes, that's right. So this is the fourth and final poll we'll be running today. So audience members, please go ahead. You know the drill. Select an option and then hit submit. So this question is, do you use CRO support for biosimilar analysis? And the options you have, yes, we make extensive use of CROs as we do not have in-house analytics. You've got, yes, we use CROs for most work, but also have a low level of in-house analytics. Yes, we use CROs for some work, but also have significant in-house analytics. And the last one is we do not use CROs as we perform all analyses in-house. So please go ahead and select whichever one applies most to you and hit submit. Richard, do you want to talk a bit more about this one just to give our audience a bit more time to vote here?
Sure, yeah. Again, I think this is going to be interesting to see how people are coping with the requirement for the huge amount of analyses that are needed in terms of the biosimilar requirements. There's a lot of different techniques that are required, a lot of different areas to study, as we've talked about today. And it's not necessarily easy for all of those to be present in-house. So I would suspect that people will use CROs for either most or some of the work, but have in-house analytics to allow them to progress work at maybe a lower level, but not to do the full high-end comprehensive structural characterizations that we're talking about here.
Okay, let's see if that's the case. Audience members, I'm about to show you the results. Thank you very much for taking part in all of today's polls. And also, I'd like to take this opportunity to remind you that we will be having a live Q&A session in a bit. So please go ahead and submit your questions if you have any. That's the box at the top left-hand corner of the screen. Right, let's look at the results from this poll. And it looks like the majority of people voted for yes, we use CROs for some work, but also have significant in-house analytics. What do you think about that?
Yes, it makes sense. Nowadays, obviously, it's quicker to have core techniques in-house to allow rapid analyses from which decisions can be taken. But like I say, because there is a requirement for such a broad range of analyses to be performed, ultimately sending those out to a third-party CRO for the more detailed work is really what I would expect to happen. So that's not too surprising, really, not too surprising. Okay, so if we move on now, just to kind of wrap things up a little bit. We've talked a lot about the structural data, and obviously there's a huge emphasis that is placed on that. But the structural data has to be contextualized as well. So we need to consider really the totality of the evidence. That's what the regulatory authorities require. It's important to get the data assessed by scientists who do have experience within this, so that what one particular technique is saying can be assessed and verified by another technique, or further work can be suggested to support or refute a particular theory that is brought up by a particular set of analyses. Of course, it's important to know about what your techniques can do. You need to know your experimental variation. If you do see a difference, you need to know whether that is down to the experiment itself or whether that's down to the sample. So you must know whether any minor differences are classified as basically not significant because they're within the experimental error, or they're significant because they're outside the experimental error, and that can lead to further work. The structural data should be assessed alongside the functional data. Obviously, if you see a difference, you need to know whether that difference is clinically active. And this is really the key. Biosimilars are not expected to be identical. They are expected to be identical enough that there is no clinically significant difference in terms of the biosimilar and the innovator. So the functional data needs to be assessed alongside the analytical data to produce a full package, a full understanding of what the biosimilar is actually capable of and what it looks like. So in summary, an assessment of biosimilarity does require very detailed, in-depth structural characterizations covering both the primary amino acid sequence, all of the post-translational modifications that are present, as well as an assessment of the higher order structure and the aggregation of the molecule. There is an expectation that state-of-the-art instrumentation and orthogonality of experimental design will be performed to generate, as I've said before, a very strong, robust package of data that will be accepted by the regulatory authorities. It's important to have a thorough knowledge of the ICH Q6B guidelines, as we've talked about today, but there are product-specific applications to that, and a knowledge of those is essential if you're going to fully characterize your molecule. As I mentioned before, at BioPharmaSpec, we do have a lot of experience across a range of biosimilar products, more than 50 biosimilar products now, and that number has gone up, as well as 150 or more biopharma products too. And we do offer a consultancy service as standard to allow you to contextualize your data within a product development program. And with that, I will finish. This is just some information about me. If you do wish to contact me, if you have any queries or anything about what we've discussed today or any requirements, then you can call me or email me with that. The information is on the screen there. Alternatively, we have a business development team that you can contact. Our head of business development is Lindsay Farrell. She's based in the UK, but we do have agents globally covering Asia, the Americas, and India. So if you wish to speak to someone locally about any requirements that you may have, then please feel free, and someone will be happy to help you. And with that, I will finish. Thank you for your attention, and I'll be happy to answer any questions.
Thank you very much, Richard. Yes, the audience have been sending in questions throughout the presentation, so I'm going to start asking these. Audience members, if you do have any other questions you'd like to ask, please do submit them now. You can do that by typing your question into the box there at the top left-hand corner of the screen, and don't forget to hit submit. Any questions that we don't get to live will be dealt with offline very soon after the webinar. So this first question is, this person says, you have explained the need for amino acid sequencing and how it is performed, but can this sequencing analysis be compromised by post-translational modifications or not?
That's a good question. Yes, post-translational modifications, obviously they're present throughout the molecule depending on what the molecule is. They don't actually pose a problem to sequencing. A lot of post-translational modifications are not present at the 100% level, so it's certainly possible to find peptides that are not modified to allow sequencing to take place. The most common type of post-translational modification that people come across is glycosylation, and particularly N-glycosylation. We've seen how many people are working on monoclonal antibodies, for example. That's an N-glycosylated molecule. Now, the site of attachment, the asparagine at the site of attachment, will not show as a cycle or is not readily identifiable out of something like a mass spec fragmentation. But what you can do in an instance like that is to deglycosylate the molecule using PNGase F. That asparagine is converted to aspartic acid. If you sequence that deglycosylated peptide, you can identify aspartic acid at that position. If you confirm that, if you then go back to the data from the non-deglycosylated peptide and confirm that it is a glycopeptide and you can see it as such, then you know originally you had an asparagine at that site and therefore you have confirmed that amino acid, because the only way that the conversion will take place to aspartic acid is if PNGase F is actually active. And that can only happen if there's glycosylation at the site, which can only happen if originally it was N-glycosylated and therefore an asparagine.
Okay, very good. Thank you very much for that answer, Richard. Just looking at the time now, and it looks like we are just about out of it, in fact. So I'm very, very sorry to everybody who submitted a question today who we haven't got to. We could only take one of them, but all of you guys will get a response. So those questions are going to be forwarded on to BioPharmaSpec, and they'll get back to you very, very soon. So thank you very much to all of you for those questions. And really, that just leaves me to thank our speaker, Richard, for what was a great presentation. And, of course, thank you very much to BioPharmaSpec for sponsoring this session. All attendees, you will receive an email shortly, and that will tell you how you can access the on-demand version of the webinar. Or you can access that's on our website as well. So that's www.business-review-webinars.com. And we do look forward to sharing further webinars with you. So if you'd like to just keep an eye out on that website, you can also follow us on Twitter at BRWebinars for daily updates. And feel free to join the LinkedIn group as well. We have that Business Review Webinars on there as well. Thank you once again, and I hope you all have a lovely day.